Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. Today, I'm delighted to be joined by Mark Lees, who is the Senior Product Manager for BioPhase with the BioPharma business unit at Sciex. And today, we're talking about the new BioPhase 8800 system, which was launched a few weeks ago and which had the pleasure of attending the press conference at and saw Mark actually presenting virtually about the new product. So, Mark, it's lovely to meet you. How are you?
I am very good, Rizwan. How are you?
I'm very well. It's lovely to meet you, and thank you for getting up early to talk to me here. Was you're calling from what is normally sunny California? Hopefully, still sunny this morning for you.
It is, yeah. The sun's burning off the clouds, so it's the cloud cover. So it is going to be a sunny and I heard a hot day.
Well, lucky for you, it's a very variable day here in Surrey in the UK today. And actually, ironically, viewers, I took out a Dodgers jersey and put it on my door just to show Mark, we're talking to someone from California. Now he tells me he's a Red Sox fan. So that's a downer straight away, but never mind. Anyway, Mark, it's lovely to meet you. Before we start talking about the BioPhase 8800 system, would you mind giving viewers a quick overview of your own background and your role? And also about Sciex as well, for those not familiar with the organization.
Sure. So I've been in the analytical space, really specifically around capillary electrophoresis for a number of years. I actually began with Beckman Coulter back in the early 2000s and have been responsible for the P800 platform, which is effectively a very well recognized tool in analytical characterization processes. We did transfer the CE business over into Sciex back in 2014. Right. Both Beckman Coulter and Sciex were or are Danaher companies, Danaher operating companies. And so it made sense to incorporate the analytical technology that we had with CE and incorporate that into the portfolio that Sciex had. And so for the last 6 or 7 years, we've been integrated into Sciex, which is really a company that has a long history, very well known for mass spectrometry and application of various types of mass spec in a broad range of industries. Our focus with capillary electrophoresis really is around incorporating this technology into the biopharmaceutical space, where it's been used extensively now for close to 20 plus years, I'd say a little over 20 years, for characterization of product purity, for product stability, product heterogeneity, as it pertains to commercial molecules or commercial drugs.
Right, that's really interesting. One thing I was going to ask you though, because you mentioned CE there, I've spoken to a number of people recently about CE. CE seems to be a lot more in focus than it was previously. Is there a reason for that?
Well, I think, it's been around, as I had mentioned, it's been around for quite a while. It was probably, when you go back over 20 years or so, it's probably been more associated with small molecules in things like stereoisomers or enantiomers, and then nucleic acids, right? So the Human Genome Project. Right. A lot of that work from a sequencing standpoint is based on the principles of capillary electrophoresis. And so there's a long history there from the standpoint of those types of molecules. The past, say, 15 to 20 years, there's been this surge of different kinds of molecules, large molecules, right? Like antibodies. And now in the last 5 years, probably an even bigger surge of non-antibody types of proteins that have been really starting to make it into pipelines and ultimately to hit the market for a variety of conditions. And so there's been a lot of emphasis on capillary electrophoresis really in the last probably 10, 15 years for that growing segment of protein molecules, protein therapeutics. And
Right.
so, the real benefits that you're seeing is that you're addressing challenges that were really hard previously, right? Getting very high resolution separation to identify very discrete entities that may be associated with a molecule, maybe a product related impurity or instability or heterogeneity, or even process related instability. So it's become a real important tool in helping define the overall manufacturing conditions and even identifying the appropriate molecules to take forward in the manufacturing process.
Brilliant. Well, that leads on quite nicely, actually, to my opening question, which is really, what is the BioPhase A800 system and what customer challenges and needs does it address?
Yeah, so the BioPhase is the evolution of our existing CE technology. And so again, we have a long history, close to 30 years of commercial CE. And over that time, we've developed systems that have effectively addressed the challenges that have come up on a day-to-day basis. One of those challenges has been relatively difficult, and that is to take the technology from sort of a low to maybe medium throughput technology into a higher throughput space, right? Sure. And that means that so instead of limiting the number of samples, for example, that you're able to analyze per day, the BioPhase is an evolution of that process to now essentially create a multi-analysis or a multi-sample a platform, allowing you to do 8 samples in parallel, right? So it's an 8, the cartridge that's associated with the instrument is actually 8 capillaries. Right. And therefore allows you to essentially, in theory, get an 8-fold increase in throughput. And so now, instead of taking days to analyze, say, 100 samples, you can do this in half a day, but not give up some of that high quality data that you're depending on to move forward in your development and ultimately commercial process.
Right. Okay. So what applications are available on the BioPhase A800?
So in its current version, we recently launched commercially. And so in our first phase of this product, we're providing a couple different applications. One is a CESDS assay, which is effectively an automated quantitative version of SDS page. Technology, which is used for protein sizing, for example. Right. In the case of CE, it's really about looking at purity, right? Looking at a particular molecule that you have in development, that you're taking through your development or your process to essentially achieve a manufactured product that you can then market for a clinical condition, and to ensure that it is what it's supposed to be, right? So the purity of that product is appropriate for the starting material that you expect it to be, right? So ultimately, what you're doing is you're separating based on size, but you're getting the resolving power of the technology to look at different entities that may be contaminating or not, or should be there, right? You may expect to have some kind of additional material within your preparation.
Right.
The other application is capillary isoelectric focusing, right? And that's the acronym there is CIEF. And this is a similar principle, right? High resolution separation, but the separation process is based on charge, right? So looking at the inherent charge of different isoforms of a molecule and understanding how that actually is affected from a stability standpoint, right? If you have thermostability or photostability, formulation buffers and things like that, that may actually lead to changes in the overall charge heterogeneity. CIEF can be used to query that.
Right. Okay. And you may have touched on this a little bit, I think, bit earlier, but can the BioPhase 800 speed up method development? And if so, what examples do you have to show that?
Yeah. So to date, so up until now, with a conventional, single capillary CE, everything's done sort of in tandem, right? You do one sample, and you do another sample, and you do another sample, and so depending on how many samples you have, that's how long it's going to take you, based on the cycle time of the assay. Here, you're able to do one of a couple of things. One is the obvious one, right? You have 8 capillaries, you get high throughput. And now you're effectively increasing your throughput by 8-fold. But the other thing you're able to do is parallel process, right? You're able to do up to 8 samples simultaneously. So it's not only about the throughput; it's about being able to do a test where you're looking at 8 different conditions, right? Eight different buffer conditions. In parallel, without the variable of time associated with the samples being on the instrument.
Right. So there's basically different chemistry conditions you can do at the same time then.
Yeah. So the instrument itself, you're able to regulate some conditions or some specifications on the instrument. We do have a single power supply. We do have a single a single buffer capability, right? Where you essentially have to match the buffer in all your samples and on your inlet and your outlet side of the capillary. But where you can actually look at differences is in your sample, right? You can look at how samples behave in different formulations. You can look at how samples behave under different stressing conditions. And you're able to then characterize those simultaneously by using that array of capillaries.
Right. Okay. And how difficult is it to transfer method from a PA800 plus to the BioPhase 800 system?
Yeah. So over the years, many organizations have built up methodologies, and they've validated methodologies on P800 plus, right? And so I've always thought of change as being something that's sort of shunned in the industry, right? You want to have consistency in process. And so we've taken that into consideration here. And although you're not able to just take a method from the P800 plus and drop it into the BioPhase system, you are able to match the specifications. You effectively would rebuild that method, but the process to do that has been simplified, right? We've created a drag and drop menu to be able to build methods, to be able to build sequences, and you can match what you're doing on a P800 very easily on the BioPhase. It isn't a copy and paste strategy, but you can actually leverage all of the different conditions that you had access to in the P800 on the BioPhase.
Right, and that just makes me think about one other question, which is that obviously all companies like yourselves have got your own software now in place. So does this use new software, or is it using existing software that you've already had out there previously?
Yeah, so that's actually a pretty good question that I want to address here. We actually rebuilt the software for this platform. In the past, we've been using a software that's called 32 Carat. Very powerful software. There was quite a few people that did use it. There were others that didn't use it. They essentially took the data files and exported to a different software analysis system. Here, we actually rebuilt from the ground up a BioPhase software package, which allows you to create methods, create sequences, then to either directly import those onto the instrument. The instrument does have a touch screen, so it has an interface physically attached to the front of the instrument, which is accessible to users to be able to download methods or sequences from either a workstation or even a networked location. And then to be able to operate the instrument directly from that touch screen interface. And so all of that together collectively make up the new software package that we've designed. We are working on some external drivers that will allow a more seamless interface with some of the chromatography data systems that are currently used by quality organizations around the world for a variety of reasons. So we are working on improving the access to external types of analytical software packages.
Great. And what do you see the future for the BioPhase 800 and the technology you're using? I mean, how do you see that developing going forward, and what the future applications could be for it?
Yeah, so we currently have the two applications that I mentioned, the CESDS and the CIEF. We are intending to, in short order, to release the glycan application. We have a fast glycan application, which effectively allows you to analyze and get glycan ID for end-linked oligosaccharides. And these separations are literally 10-minute separations. And then we'll integrate into an ID database, which will then allow for glycan IDs to be determined. We're working on an RNA assay. We're working on some viral vector assays. So lots of stuff coming down the road here, from the standpoint of what the current. So we're really transferring existing validated assays that we had on the P800 Plus, or that we have on the P800 Plus, but we're adding some of these new assays, which are going to be really addressing some of the new challenges that are coming up in the industry today. And so, I always like to. It's been more difficult with COVID, right? With the pandemic situation, because travel has been limited and so on, and going to trade shows hasn't really been a thing for a while. And so, I like to go out and really understand what's coming. What are the coming attractions? What kinds of molecules are being addressed or added to pipelines? And then, really, the question is, how can this technology help make developers' lives easier by really exploiting the power of the separation that we're able to achieve?
Brilliant. Well, that actually leads on to almost my final question, anyway, which is looking ahead then. And you mentioned trade shows there, and I know Sciex had a presence, I think, at the Biotech Week. I think it was Biotech Week. I might be wrong for the shows, but a couple of weeks ago. But so will you be at ASMS, and what other events will you likely be at where people can get to see the new instrument I was going to say in the flesh, but you know what I mean.
Right.
And touch it and feel it and see the demos for it.
Yeah, so we're still working on our schedule. Obviously, it's a little bit in flux right now because we're trying to figure out who's having a live show, who's not. We've had a lot of virtual activities. So we were at the CE Pharma meeting, which is basically the application of CE in the biopharmaceutical industry conference, which is a big industry conference specifically related to CE technology. So we did have a virtual presence there because it was in fact a virtual conference. It was run by the CAS organization. But as we start to see more conferences go live in person, you'll see us attending these. We will be at ASMS, but that's primarily a mass spectrometry conference.
Right.
So there will be, obviously the presence of some of our mass spectrometry technology. Our new Xenotoph instrument, for example, will be highlighted there. And so, because of that particular, the crowd that would attend that would be more interested really in mass spec. We're probably not going to have the BioPhase highlighted there. But as we move through the rest of this year, there are some conferences, and then WCBP, which is the Well-Characterized Biologics Biopharmaceuticals Conference in Washington, D.C. in January. We would intend to be there as well. And obviously it depends on if it's live or not, right?
Yeah, everything's in flux, as you say, sure.
Yeah. So, I guess my message is keep your eyes open, and if it's live, there's a good chance we're looking at attending.
Fantastic. And obviously outside live events, where can people get more information?
Yeah, so probably the best place to go would be the SCIEXCE.com website. And then if you basically follow through to the CE, the section on CE or capillary electrophoresis, you should be able to get all the details on the BioPhase platform within that portal.
Brilliant. Okay, well, I'll dig that out for you, so don't worry, you don't have to search for it. I'll find the link and put it above the video, so you don't have to, you just have to click. So there we go. So,
all right, great.
All right, well, Mark, it's been lovely talking to you. Thank you very much. I don't know how Boston, the Red Sox, are doing for the wild card place at the moment. Because I'm also a semi-Yankees fan as well, so I'm not sure what the situation is. But I think the Rays have got it all set up anyway. Like, in all honesty, in the East. But it was lovely to meet you. I wish you lots of success with the rollout with the BioPhase 8800. I'm sure it'll be a great success.
Thank you. It was good talking to you too.
And it's lovely talking to you. And viewers, if you've got any questions for Mark, feel free to leave them in the comments below, or you can send him a LinkedIn message. He's on LinkedIn. And check out the links for the BioPhase 8800. It will be above the video, so you can actually click on the link and find out more as well. And as I said, as Mark mentioned already, look out for SCIEXCE at upcoming events. It is a very, very difficult situation for both organisers and exhibitors at the moment, as we all know, because things are constantly in flux. But obviously, if they can be, they will be there, and you'll be able to see the product there as well. So all that's going to say is, Mark, thank you very much. Enjoy the rest of your glorious sunny day in California. I'm very jealous. Thank you very much.
All right. Thank you, and have a good weekend.
You too. And thank you, viewers, for watching. And until next time, as always, stay well and stay safe. Bye-bye.